Okay, so this is going to be a video for Wednesday. Our goal is to finish all of the stuff about the cell cycle and DNA by the end of the week so that we can do a little bit of new stuff over break. So this is just the cell cycle. um, Cells have to reproduce or else they die. All cells have like a certain lifespan depending on the type of cell. The cell cycle is divided into distinct phases: G1, S, G2, and M. I would list those in your notes because we're going to go over each part of the cell cycle specifically. They they have like this state in which they are not doing anything. It's called the G0 phase, where they are not dividing at all. That can also happen. So G1 phase, also the diagram on the right is really great. During this time, organelles are reproducing, protein synthesis is occurring for growth and for differentiation. The cell is not technically reproducing, but it is preparing for duplication. So things are doubling so that when the cells split in mitosis, they have enough of the material in both. Because transcription is occurring, DNA is uncoiled. Transcription is the making of proteins, and I need those proteins to make more organelles and things like that. This phase is really variable. It can range from very short amounts of time, depending on how many cells I need, to years. But you can see under G1, G0 is not even happening. And so some cells can be in that like static state for a very long time, G0. G1 stuff is happening, it's just not splitting yet. The average time is about 10 hours if the cell cycle lasts for 24 hours. So it is the longest stage. Most cells that are going to differentiate will do it during this phase as well. So whatever is being produced, it assumes it's like correct identity. And so if the cell is not reproducing at all, it's not making any of that, it is in G0. Nerve and muscle cells usually are in G0. They don't reproduce the same way that we do, or that cells that are in G1 do. So that's the G1 and sometimes G0 phase. This is the S or synthesis phase. It is the second phase. DNA uncoils. Usually it's already uncoiled because we're making proteins. DNA is going to replicate because I need a new copy. Any additional organelle replication occurs, so I have enough organelles for both of my new cells. And this phase ensures that the daughter cell has the same DNA as the mother cell or parent cell. The G2 or GAP2 phase, this occupies time between the creation of DNA and actual mitosis, which is the division. During this time, the cell prepares for mitosis by making and organizing proteins, such as tubulin and microtubules, that are used to physically create those like spindles that come together and then split the cell apart. And so G2 is like the prep for the actual division. During M, or the mitosis phase, the nucleus is replicated and the cytoplasm divides to produce two genetically identical cells. These phases are triggered by certain signals, and if those signals don't happen, we're not going to have those things occur. The amount of DNA varies during the cell cycle. This graph represents how much DNA is present. Obviously, we see that G1, it has its normal amount. In S, it is increasing. By G2 phase, it is totally copied into twice as much DNA in the M phase, but by the end of the M phase, when the cells have divided, it's going to be back down to its normal amount. There are some internal controls on the cell cycle. We have this accumulation of chemical signals that will allow cell division to occur or not occur. Oftentimes, they have to be phosphorylated, or they have to be attached to a phosphate to work. Think about this in terms of cancer as well. We know that in cancer, the cell cycle is not functioning, and a lot of the time, it's due to the incorrect cell signals. Two types of signals are your cyclins and your kinases. Cyclins are proteins that control the progression of the cell through the cell cycle. And so it's a chemical that does that. Kinase is a type of enzyme that transfers a phosphate group to a molecule. It does the phosphorylation, so it activates cyclins and the molecules that are going to control your cell cycle. Cyclins are made during times during the cell cycle to push the cell cycle to continue or stop. They are destroyed after they're not needed anymore. There's also 4 different types of cyclins, and so we see that right before and during each phase, there's a big upswing of cyclin D occurs the whole time, cyclin E right after G1, cyclin A during G2, B during mitosis. But these chemicals always push they always push the progression of the cell cycle. CDKs, kinases, they're the enzyme, and so they don't get destroyed, whereas cyclins do, because we need to cut off this cell division at a certain point. So when we talk about the idea of a kinase, an enzyme that phosphorylates, they have 2 different forms. Usually, hi, and we're back. So certain kinases have 2 forms, either active or inactive. They're the enzymes that phosphorylate certain things. Most of the cell signals are phosphorylated by a kinase, and the kinase has to be active to phosphorylate things. Most cell cycle kinases are activated by the molecule called a cyclin. So the sequence of events would be a cyclin shows up, it turns on your kinase, and then the kinase phosphorylates the molecule that is going to push the cell cycle to continue. So, and again, a cyclin-dependent kinase is going to be a kinase that requires the cyclins. So once it phosphorylates a signal that pushes the cell cycle forward, the cyclin breaks down, the CDK stops working, and so phosphorylation doesn't occur anymore. It is no longer active. Above is an example of how the M cyclin affects the CDK activity or the kinase activity. So we can see that every time I have a peak in the cyclin amount, my kinase activity peaks, and then it dips. We've seen this before as well. So we can imagine at each of these peaks of cyclins, we'd also see a peak at the activity of kinases and phosphorylation. These are all the different types of cyclins, and on the far left column are the kinases that they activate, and then it's also the roles. You don't need to know about the specific roles of each cyclin. So they control the cell cycle. We also have checkpoints. There are 3 during G1, G2, and during mitosis. These are checkpoints that everything needs to be functioning normally for the cell cycle to progress, or else it will shut down because they assume that a mutated cell is being created. There's also external factors that influence the cell cycle. For example, when there is a wound, the cell cycle is going to accelerate because we need to stop the bleeding. There's inflammation. Proliferation is the creating of new cells, and then remodeling, structuring them into the new tissue. So that's the end of that set of slides. We will pick up on the next PowerPoint.